So it's a great pleasure to introduce Nick Robakis. Nick has been at Mount Sinai for 35 years, one of the first people I met when I was thinking about coming to Mount Sinai. And we're on the same floor. Nick has been, and remains a world leader in multiple aspects of neurodegenerative disorders. Obviously, has made a major and very long-standing impact on Alzheimer's research. Was one of the early cloners of APP. Has done some work on prion proteins. Has also worked on FTD, frontotemporal dementia. Kind of looking at the molecular underpinnings of these disorders and trying to understand the functional impact of mutations in some of the genes that are associated with the disorders. I should mention presenilin. He's been a world leader also in presenilin, another major, critical, important part of the Alzheimer's genetic architecture. Has run, a very well-funded laboratory for decades and continues to advance many aspects of Alzheimer's research and FTD research, frontotemporal dementia research, and other disorders in that context. And I don't have the title of your talk in front of me, but it was, I guess, the previous slide. It's clinical trials, FAD, and clinical trials, FAD variants, and the etiology of Alzheimer's disease. So, Nick, thank you for doing this. We look forward to a great talk. And just to remind everybody, sorry, Nick, there's a Q&A button. If you have questions, just put them in there, and I will share them with Dr. Robakis, either in real time or more likely at the end. But if it's a question that's important to answer right away, I'll try and if I see it, I'll share it with Dr. Robakis. Thank you, Nick.
Okay, Joe, thank you for a very, very generous introduction. I really appreciate that. And I just want to mention that I gave a similar lecture about a year ago in neuroscience. So, so anybody who heard that lecture is not going to learn much more from this one. So this is about Alzheimer's disease, the neuropathology. All of you know that, as all of you know, it's characterized by amyloid plaques, extracellular amyloid depositions, and intracellular neurofibrillary tangles. Those are the two hallmarks, the two pathological findings that Alzheimer himself observed more than 80 years ago, and still for the disease that still bears its name. And we still use clinical dementia plus the neuropathological hallmarks of amyloid plaques and neurofibrillary tangles to diagnose Alzheimer's disease. What we have to have in mind as we go through this lecture is that AD is really a disorder with clinical, genetic, and neuropathological heterogeneity. And that is a very, very important issue to be discussed. So, the main reason that it took about 80 years to elucidate the molecular nature of the amyloid plaques. Amyloid plaques, by the way, it has attracted most of the attention as an etiological agent of Alzheimer's disease. And it took about 80 years to clarify the chemical structure. And the reason is, of course, that amyloids are very insoluble in detergents that we usually use chemically to dissolve. And it was in 1984 when George Glenner used formic acid, and he was able to dissolve the amyloid, and he discovered that it consists of peptides, about 42 amino acids long, or 40. They have really not well-defined ends. And I just want to mention that the two peptides that are most common in the amyloid is the one 42 amino acids and the other 40 amino acids. And these peptides, they aggregate to make fibers or of beta pleated sheet structure and precipitate out of solution. So in some like 35 years ago, several people following Glenner's discovery, they were able to isolate the gene that from which this peptide derives. And the gene is a very famous protein today. It's called APP. It encodes APP. And you see really that the amyloid fibers, the amyloid peptide that aggregates to form the amyloid fibers, which are being deposited not only in the neuropil as amyloid plaques, but also as cerebrovascular amyloid. It is a small peptide in a small fraction of the APP protein, which is much longer, about anywhere between 695 and 770 amino acids. And under certain circumstances that we don't still understand very well, this peptide aggregates from the soluble form. It forms the fibers and precipitates as amyloid plaques or cerebrovascular amyloid. Why these A-beta peptides are in all of us? They are not being really made only in the old age. They are there from the time we are born. It's been made by a lot of different cells. But we never see amyloid in young people. So somehow the peptide forms fibers and precipitates in the brains of older people and Alzheimer's disease people. Now, what's the mechanism that pushes this precipitation? We don't know yet. So that is a very good question. Why the A-beta peptides that are soluble and are present in all people, they only precipitate in aged people and in AD people and Down syndrome people also. So, and now that we said Down syndrome, APP was shown to be part of chromosome 21, the trisomy of which forms is responsible for this genetic mental disorder of Down syndrome. And this now aged Down syndrome people have very similar neuropathology as Alzheimer's disease. And part of it is, we know now, it's because APP is on chromosome 21. So, but the central question of Alzheimer's disease is what causes the accelerated neurodegeneration that leads to dementia? Because Alzheimer's disease is, first of all, a neurodegenerative disorder. Neurons die in the cortex and subcortical regions of the brain. And you see here an example of pyramidal cells, cortical neurons, how much can be devastated in AD brain compared to normal brain. So especially some categories of neurons are being devastated by the disorder. So that is the central question in Alzheimer's disease. What causes the accelerated neurodegeneration that leads to dementia? We still don't know what's going on here, although we have lots of theories about what's going on. So, and as a result of the neuronal death, you can see also that the mass of the brain shrinks, and this is part of today's efforts to diagnose Alzheimer's disease using brain imaging. So very briefly, just for the history, to go a little bit back and see when this question was first asked, what were probable answers. So the first to make an amyloid, to blame the amyloid deposition for the neurodegeneration, and I was to say that the amyloid fibers deposit in the brain, and they form the damage, and they cause the damage. A very logical explanation was Glenn's theory in 1966, where he says that the A beta peptide is not being made in the brain, but rather in the liver, in the periphery, and comes to the brain through the circulation and gets deposited in the brain vessels, in the cerebrovasculature of the brain. And this deposition weakens the walls of the vessels, and slowly we have leakage of serum into the neuropil. Serum comes in contact with neurons, and that's what starts the neurodegeneration. As you see, the immediate, the proximal, according to Glenn and according to this theory, cause of the neurodegeneration is the serum that comes in contact with the neurons, and because serum can be toxic, it starts the neurodegeneration. Now, the plaques, according to this model, are formed from the serum that comes into the neuropil, and then the A beta peptide precipitate in the intracellular space. So, as you see, the role of the amyloid in this theory was just to break the blood vessels and to breach the brain neuropil barrier. So, but when it was discovered that APP was made by brain cells, and the A beta was not necessary to invoke the circulation, it was clear that A beta was made by brain cells, so that it could be that brain cells were making A beta, which was again precipitating as amyloid. And the theory, a second theory, was one that was promoted by Selko and Hardy in this paper in 2002. And the central point of this is that the amyloid that deposits in the neuropil is toxic and produce toxicity to the neurons, which make neurofibrillary tangles and cause Alzheimer's disease. Now, this theory was again revised by other workers, and they propose that it's not really the plaque amyloid, but the soluble 42. As you remember, 42 is one of the more abundant A beta peptides, and because it's more hydrophobic than 40, it is more toxic, and it forms oligomers, is easier than the 40. So when we have more increased amounts of 42, it favors precipitation and formation of fibers. So they say it was the soluble A beta 2, or its oligomers of mainly A beta 42, that they act as the proximate toxic factors in AD. So, so that was the third theory, that oligomers, A beta oligomers of 42 or mostly 42, and even the ratio of 42 to 41 when it increases, it's also a risk factor. So it's important to understand, as we said, that Alzheimer's is a heterogeneous disorder. And we have at least two forms of AD, probably much more many than two. But the sporadic AD and the familial AD. Most of the AD cases is of sporadic form. And we call it sporadic because they have no obvious genetic etiologies. But does not mean that genetic factors do not play a role. Of course they do. And then the familial AD has very simple Mendelian mode of transmission. It's usually autosomal dominant. It's a specific mutations in 3 proteins. 2 are presenilin 1 and presenilin 2, and also the third protein is APP. So it's amazing, presenilin 1 especially is the most interesting one of the 3, because there is a very large number of missense mutations that all cause Alzheimer's disease. Now, it is out of the about 450 amino acids residues that presenilin 1 has, almost half of them, 250 or something, or more than half, can cause Alzheimer's disease when they are mutated. So all these variants of presenilin cause the FAD form of Alzheimer's disease with autosomal dominant transmission. The APP also, about 30 different mutations have been found that are very strong risk factors for Alzheimer's disease. And that, of course, was an additional argument for the amyloid theory, that amyloid is indeed the risk factors or the toxic factor, a pathological factor that causes Alzheimer's disease. Now, it is interesting to say that the 2 proteins kind of collaborate to form amyloid beta peptide, because presenilin cleaves APP at this point here within the membrane to form the C terminus of the amyloid peptide, and beta secretase, on the other hand, cleaves the APP and the N terminus of the A beta peptide to excise the A beta, which then precipitates. So it looks like presenilin and APP were working together to form A beta peptide. So that's a little bit strange. Why would you have a system like that in nature to make amyloid? Unless the amyloid beta peptide has a very good biological function, it looks a little bit counter intuitive and not very well fitting into evolution. So as we'll see later on, there are other very important cleavages here, and we're going to talk this at the end of my talk. So, but it looks like the field was concentrating on the positive factors that were promoting amyloid as a cause of a disease, but we were somehow overlooking that there were lots of strong arguments also against this theory. And those arguments are summarized in this perspective on disease that we published in 1998, which we cautioned that it's not going to be so straightforward, and amyloid may not, after all, be the main causative agent of neurodegeneration. And that was 23, 24 years ago. And that was because it looked like the field overlooked evidence like the poor correlation between brain amyloid and plaques, the degree of dementia, the neuronal loss, and even the formation of NFTs. All of this did not seem to correlate well with amyloid, and an etiological factor has to correlate with the intensity of the disease and the location of the disease. But amyloid did not really fit this bill. So the other thing was that a lot of normal aged people, about 1/3 of all aged people, they have similar amyloid plaque levels as the AD patients, but they were fine. So that was a problem with the amyloid theory. The soluble A beta theory had the problem that we could not really isolate, detect an AD specific oligomer that was specific to AD. And we could not even see a specific and consistent increase in any of the oligomers that could be segregated with AD. So really we were saying oligomers were toxic and may cause AD, but upon examination of the brain fluids, we could not find an oligomer that was specific to AD. And also, mouse models, they showed of amyloid, they showed little neurodegeneration. So that was evidence against the theory. But anyway, and also the theory that all the FAD mutations increase the A beta 42 or the ratio, it turned out that was a little bit exaggerated, because some FAD mutants really increased the 42, but others did not. And that's what we reported in this paper back in 2007, that although some mutations increase A beta 42, some others do not. And that was a paper that later was replicated by a much larger study where they use something like 130 FAD mutations of presenilin, and they found in essence the same data as we did, that although some FAD mutations do increase the A42 and the ratio, many others do not. So there was an imperfect correlation or linkage. The other thing that we should bear in mind is that genetics may indicate a genetic locus where targeted by a disease, but in this case, for example, amyloid and presenilin, but it says, I mean, APP and presenilin, sorry, but it doesn't really say much about the mechanism. So the presenilin and APP did not only make amyloid, they had other biological functions. And those biological functions, we really failed to pay attention to, because both proteins, they have a lot of very important signaling functions in the brain. And that's what we're going to talk a little bit later on. So in any case, the amyloid theory was put to test, starting with a very nice experiment that Dale Schenk performed in 1999, where he was experimenting with amyloid in mouse brains, and he observed that immunization with anti-A beta 42 antibodies could clear amyloid. And that fundamental observation finally led to making antibodies that can clear the amyloid. And the second advance in the field was to make compounds that could bind to amyloid in vivo and being used in PET scans to follow the amount of amyloid in the brain. So the antibodies could clear the amyloid, and the PET scans can tell us how much amyloid was cleared. And as you see here, the Pittsburgh compound, the so-called Pittsburgh compound, a thioflavin chemical that binds to amyloid, could be used in PET scans to measure the amount of amyloid in the cortex. So using that, it became possible to use patients and to clear the amyloid and see what does it do to cognition. In addition, because the A beta is being made by the cooperation of at least two enzymatic systems, the gamma secretase and the beta secretase, the field made pharmaceuticals agents, chemical agents, that could inhibit the beta secretase or the gamma secretase. And by inhibiting the beta secretase, this cleavage here, or the gamma secretase, could inhibit production of A beta. And in addition, amyloid depositions in the brain of AD people could be cleared by immunoprotocols, as we said. So the problem was that removal of amyloid plaques or inhibition of the production showed no benefit to AD patients. To make a very long story short, all the antibodies, none of the antibodies against the the A beta depositions showed cognitive effects. So that suggested that probably depositions may not be the best therapeutic target. So, but still, there was this theory that still amyloid could be the agent, the etiological agent of Alzheimer's disease, the main agent. But because we measure AD people, we put in cohorts, AD people that already had dementia for a long time and lots of neurons already have died, we could not now, it was damage done, and because neurons do not being replaced, we could not have improvements. But really, later on, MCI people, mild cognitive impairment people, people that were not on this stage of dementia, but much sooner, where there was lots of cognitive function still, and lots of neuronal populations were intact. So when companies and academic institutions use MCI cohorts, patients that they were only mild cognitive impairment, and they apply the drug application, still would not see improvement. So ideally, of course, you would expect the drug effect to be parallel to the normal agent. Of course, that nobody could expect that. But everybody was expecting to see a decrease in the rate of deterioration when the anti-amyloid agents were applied all the way in MCI. But we did not see that either. Instead, they followed the same deterioration curve as the yellow one, which is for the Alzheimer, for the controls, the Alzheimer patients. So there was really, so the argument that this is damage done really doesn't seem to be very strong. So in addition to the amyloid, the second neuropathological neuropathology that the agent, the hallmark that is a candidate and has been used to make drugs is the NFTs, the neurofibrillary tangles, which really consists of tau protein. The tau protein is a MAP protein that functions, a microtubule associated protein that functions in cellular transport. And there are at least 6 different isoforms of tau. Most of them are present in the NFTs, the neurofibrillary tangles, which deposit mostly around the nucleus. So the approach is now to get rid of this hallmark, pathological hallmark, is to inhibit tau phosphorylation, because the theory is that tau phosphorylation increases the chance, the probabilities that tau will aggregate and form neurofibrillary tangles. Of course, one way is to use kinase inhibitors. Kinase inhibitors will inhibit tau phosphorylation, or to use inhibitors of aggregation, or to use the same methodology as for the amyloid plaques, in other words, antibodies against NFTs to clear the formation of antibodies. So now tau is present in many different brain disorders. Lots of brain disorders that are shown here, indicated here, have as a hallmark abnormal tau precipitations. So we call them all together tauopathies. So the tauopathies can have genetic etiology, like the frontotemporal dementia and the familial AD, but also environmental, like the dementia pugilistica, the chronic traumatic encephalopathy, which clearly are environmental. So the TBI, the traumatic brain disorder, I'm sure all of you heard about that, and the chronic encephalopathy are due to brain trauma. And from that, we have not only neurodegeneration, but also sometimes amyloid formation, almost invariably formation of neurofibrillary tangles and inflammation. So this shows that amyloid depositions and abnormal tau depositions and chronic inflammation can be the result of brain toxicity, of a brain toxic factor, in this case, the TBI. So in other words, they could be the result of neuronal stress. The trauma stresses the neurons, and in response, the neurons can make amyloid or neurofibrillary tangles of course, inflammation. So the question really is, although the etiology is environmental, the question is, can we, however, improve cognition by dissolving the NFT depositions? And can we stop neurodegeneration and dementia by getting rid of NFTs? That's really the question concerning NFTs. But it looks like the question is, the answer is not straightforward. And this is because in AD, neuronal loss exceeds many times the number of NFTs that indicate that many neurons die without ever forming NFTs. That's an old finding, actually. It also shows that there is evidence that conditional suppression of tau expression does not necessarily lead to improvement. And the large number of distinct brain diseases that form tau indicate that NFTs are probably non-specific cellular responses to neuronal stress. And thus far, sadly, agents that target NFTs fail to benefit AD patients. So it looks like the same story is being repeated, the story of amyloid, including a new clinical trial of Genentech, where anti-tau NFTs antibodies had no effect on cognition. So, although NFTs may be useful markers for the disease, they probably are not causative for the FAD. So, as you probably all know, FDA did approve an antibody of Biogen, but as a therapeutic agent of AD. But this was, what was really approved was the fact that the aducanumab could remove amyloid, and it did remove amyloid, but had no effect on cognition. So that was a very controversial, actually, approval. And as you know now, many other countries like Europe and the European Union and Canada did not approve the Biogen antibody, the Aduhelm, as a therapeutic agent. And even in the United States, now it's restricted only for clinical trials. So unfortunately, neither the tau theory nor the amyloid theory led to therapeutic strategy, to therapeutic agents. So, so what's important to understand for AD? It is a difficult disorder to deal with because it displays great clinical, neuropathological, and genetic heterogeneity, and that causes lots of problems, even with the cohorts that we can use to test the disease. So now, because there are many aged people with amyloid and tangles, but with no Alzheimer's disease, the concept of resilience was put forth to explain why so many people have amyloid levels as those with Alzheimer's, but they have no cognitive problems, or they have mild cognitive impairment. So the resilience, in essence, says that these people have something in them that makes them less vulnerable to the amyloid depositions or to tau depositions. But an alternative explanation is that none of them are causative, and the FAD mutations also affect functions of APP, functions of presenilin, functions of presenilin 2, and targeting these functions by the disease, it causes problems in the neuron, like problems in neuroprotection. So we wanted to see whether FAD mutants of proteins that are involved in Alzheimer's disease, like presenilin, may target important cell survival functions independent of neuropathology. So we first tested whether presenilin has neuroprotective functions. So since presenilin is involved with neurodegeneration, could it be that that the presenilin variants involved in neurodegeneration target neuroprotective properties of presenilin? So, and to ask this question, we first stressed neurons with excitotoxicity. Excitotoxicity, as a matter of fact, seems to be involved in neurodegeneration. So, and we asked whether how could we find out whether the FAD mutants affect this hypothetical function of presenilin. Now, to go from hypothesis to experimental test, we used primary neurons from mice, brain, cortical neurons. And we had three different types of neurons: wild type neurons, neurons that they were heterozygous, in other words, they had only one presenilin allele out of the two, and homozygous knockout of presenilin 1. So these neurons here contain no presenilin at all, these primary neurons. These neurons are heterozygous knockout, and these are normal, wild type. What we found was that when neurons are being stressed by glutamate, that was by excitotoxicity, and you hit the neurons with a brain factor, FNB1, which is neuroprotective, it protects the neuronal death. It was greatly decreased, which showed that the FNB1 acting through its receptor, it was rescuing neurons from excitotoxic death. But in the plus minus neurons, which contain only one allele or minus minus, we had almost no neuroprotection. So this was a simple but clear experiment that presenilin somehow has neuroprotective properties. And the same thing we found for BDNF. It was behaving almost identically as the FNB1. So both neuroprotective factors needed presenilin to work in protecting neuronal population. And that's in vitro. But all of this function was independent of gamma secretase. Interestingly, having one allele, it was not enough to have neuroprotective function. And this reminds us of haploinsufficiency, which it is very famous in frontotemporal dementia with progranulin. You need both progranulin alleles to be functional to have protection in progranulin. So, so we went a little bit further and asked the question, how does presenilin inhibit the neurotoxic activity of excitotoxicity? The FNB or the BDNF work through the receptors. The FNB binds to FB receptor and the BDNF to TrkB. So what we discovered in essence was that presenilin facilitates the interaction between FB receptor and NMDA. Now, NMDA receptor regulates excitotoxicity, and that's where the secret is. So presenilin apparently was a factor that was facilitating the interaction between the FB2 receptor and an NMDA receptor. And the same thing we found for the interaction of TrkB with an NMDA receptor. When you had no presenilin, this complex here was much reduced. And we follow the GLU-N1 subunit of an NMDA receptor in an immunoprecipitation experiment. It showed that the complex between NMDA and FB2 was greatly decreased in the absence of presenilin, the protein. So we then ask, is this really very important complex for the neuroprotection? Could we somehow inhibit formation of this complex and see what happened to neuroprotection? And when we did that by making a peptide that would interfere between the interaction of FB2 receptor with an NMDA receptor, we abolished neuroprotection, and you see that here. So this is the FNB1 is neuroprotective, but it loses its neuroprotective activity in the presence of this peptide that inhibits the complex between an NMDA receptor and FB2 receptor. And we found a similar paradigm for the BDNF. So it looked actually that this is a survival complex, and that when you block its formation, you block the neuroprotective activity of the FNB or BDNF. Now, what are the effects of the FAD mutants on neuron survival? We found now that presenilin, by facilitating interaction between the FNB receptor or BDNF receptor and NMDA receptor, a receptor that regulates excitotoxicity, it protects neurons from neurotoxicity. So the question was now, how do FAD mutants affect this function of presenilin? And to make again a long story short, this has been published. We found that we found that FAD neurons, in other words, that express FAD mutants, in this case the 146 presenilin 1 FAD mutant, in either homozygous or heterozygous state, they lose neuroprotective function. The FAD mutation somehow targets the ability of presenilin to be neuroprotective. And this happens in two different, these neurons are derived from knock-in mice that express those, each one express the 146 mutant, the other the 213 FAD mutant. So both neurons from both mice brains behave similarly. The FAD mutations targets the neuronal function, the neural survival function of presenilin. and they have dominant negative effects, which is very interesting because the FAD that these mutants cause is a dominant disease. So anyway, we also test the electrophysiology of an MD receptor. How is an MD receptor being affected, therefore, by these mutations? Can we see that by electrophysiology? And that's what we did. We measured the excitatory postsynaptic currents with, in collaboration with George Huntley here, Huntley's laboratory. And we found that, that mice brain slices from mice that express FAD mutants are different than the wild types. And they have much reduced response to evoked excitatory postsynaptic currents. So in summary, our data showed that FAD mutants decrease neuroprotection against toxic insult and impair also electrophysiology, NMDA receptor electrophysiology, independent of any neuropathology, because these mice do not show any AD neuropathology. So these functions were independent effects of the FAD mutations on the physiology of the cell. As I mentioned, the neuroprotection activity that we measured was independent of gamma secretase. But a very important function of presenilin 1 is the gamma secretase function. And here you see a summary of what we know, more or less, about the effects and the function of gamma secretase system. Gamma secretase, as we mentioned, we have a cleavage by base, and the base cleavage forms the N-terminus of the A beta. And then we have cleavages at this point here that create by gamma secretase, by presenilin 1, that form the C-terminus of the A beta peptides. So as we said, this makes little sense from evolution point of view. So pretty soon after the gamma secretase cleavage, it was discovered another cleavage that we call the epsilon cleavage by the group of Conrad by Reuter. And it turned out that this cleavage was much more important than initially was appreciated, because it looks like this cleavage of, in this case, APP by gamma secretase creates intracellular peptides that they go on to transmit signals. So there are signal transductions peptides. And I'll tell you, I'll go briefly over two examples of the function of gamma secretase as a signal transduction agent. This is with NATS. NATS1 is a very important receptor that regulates cell differentiation and cell proliferation. So NATS from the cell surface signals to nucleus and changes gene expression. It regulates gene expression of genes involved in cell differentiation. That signaling to go to the nucleus requires a gamma secretase cleavage. And that, which is very close to the cleavage that makes the A beta peptides in the APP. Now, as we probably appreciate now, many proteins have been cleaved by gamma secretase, and that was part of the confusion. It is not only APP, it's a large number of proteins that have been processed by gamma secretase. So one is the NATS example, paradigm, for the very useful function of gamma secretase. APP also has a corresponding cleavage that forms a CTF of APP that may regulate gene expression. And another example is the ephrins and the CBP and CREB. In essence, what happens is that upon calcium influx into the cell, that influx stimulates the presenilin gamma secretase cleavage of N-cadherin, which binds to CBP and regulates expression of gene expression of NCRE of CBP. Now, later it became out that many other proteins, as we said, are undergoing the epsilon cleavage, which then a lot of these peptides go to the nucleus for to regulate gene expression. So we ask then, how do the FAD mutations affect this cleavage, the epsilon cleavage, and not the gamma cleavage? And what we found was that the FAD mutations, a lot of FAD decreased the gamma secretase cleavage of a lot of gamma secretase substrates. And you see here how many different mutants of presenilin 1 inhibit production of the NCAD-CTF2, which is this fragment here. This is duplicates. It shows how the FAD mutants decrease the epsilon cleavage of cadherins, which was analogous with the epsilon cleavage of APP and analogous of the epsilon cleavage of NATS and other proteins. So the FAD mutants, it looks, they affect the epsilon cleavage, which the signaling cleavage mediated and catalyzed by gamma secretase. We published 2 more examples of that with the ephren B and the FB receptor. And we showed the same thing, that the FAD mutants decrease tremendously this peptide that has signal transduction functions. And because this system, the ephren B and FB system, is very much involved in brain angiogenesis, we ask the question that how do the FAD mutations affect the function of this system in angiogenesis? And that's what this shows. This is the ephren B4 and the ephren B2. Those two receptors are receptors and ligands to each other. It's a bidirectional system. And by cooperating, they stimulate angiogenesis. Now, there are lots of evidence in Alzheimer's disease of vascular abnormalities. And the brain has a process that's called neovascularization that it's used to tissue repair. And neovascularization is a very complicated process and regulated by the ephren B2 and also by the VEGF receptor, where you create new blood vessels from existing blood vessels. So this system plays an important role in this process. So we wanted to see how is this being affected by presenilin and presenilin mutations. So when we had a system to measure what we call angiogenic functions of endothelial cells, and in essence what we found is that when we inhibited gamma secretase activity, the endothelial cells also were losing angiogenic functions like sprouting tube formation and angiogenic complexes.
Nick, can I interrupt for 1 second?
Yeah.
I'm a little sensitive to time. People tend to get off fairly promptly because they have the next meeting. So there's 5 more minutes. Of course, you can go on a little longer, but I do think you'll start losing people.
Okay. No problem.
Yeah, okay.
So in essence, the FAD mutations, to make a long story short in the interest of time, were affecting the angiogenic functions of the brain neovascularization system, which was a pretty neat observation, we thought, and may have effects and the way the FAD mutants may be toxic to the brain by neovascularization. So, in the interest of time, I'll skip that because it shows actually how the blood, the work has been published in molecular psychiatry a year ago or two now, and can be reached there. But the FAD mutants were impairing neovascularization of damaged brain areas. And then we saw that also brains that were expressing FAD mutants were less able to protect neurons from death, and also they had cognitive impairments. So in essence, I'll skip these slides. I would say that our data show that presenting FAD mutants target endothelial cell functions. So it's very important, we thought, to also concentrate on biological functions that may be targeted by FAD mutants. And we propose that probably both amyloid depositions and tau abnormalities may be actually results of neuronal stress that leads to neurodegeneration and may be side products of that neuronal stress. Thank you very much. Here is my collaborators listed.
Thank you, Nick, for a terrific, wonderful talk, and for kind of a unified theory of Alzheimer's disease. You went through the last bit of, I don't see any open questions, and we only have time for maybe one. But maybe you could just clarify what you just went through quickly. So, the presenilins, if their mutations are impacting neuroprotection, right? So do you see, what about other gene mutations like APP and sortilin-1, if that's the right one? Do you think they all operate through similar mechanisms?
No, I don't expect them to operate through similar mechanisms. I think the APP mutations will operate through APP functions. Mm. And unfortunately, we know very little. It's amazing. We know very little about APP functions.
It is amazing.
And if you take into account the heterogeneity of even the FAD is very heterogeneous. I mean, the different presenilin mutations result in different phenotypes of neuropathology, distribution in the brain, and even neuronal loss in the brain. So maybe we cannot expect probably that each FAD mutation works through the same mechanism. I think that would be too much to expect.
Yeah. Yeah. Okay. Well, thank you for that tour de force, and thank you for that perspective. I think I remember well when I was a postdoc at Rockefeller, and you and colleagues discovered APP, and Sam Gandhi, who was then also at Rockefeller, began a program on APP studies that me and a couple other people joined. So that was really the beginning of a very, very long and productive series of studies by your group. And I think the disappointment, I'm sure, is that, as you said, there's still so much we don't know about some basic things like, what does APP do?
I think so. The heterogeneity, it's a big problem. This is not one disease.
Yeah. Huh?
And that's a big problem. We have to first characterize it and group it.
Yeah. Yeah.
Okay.
Well, thank you very much, and I want to thank all the participants.
Thank you.
Nick, you got 40 people who joined, even though you gave this talk a year ago. I think it's a very exciting and interesting topic, and people wanted to hear what you had to say. Okay. And I'm sorry we don't have time for questions, but see you all soon.
Okay. Bye-bye.